see San Diego, tell us about our state flower. California Mountain.
Awesome, thanks, Ben. Thanks, everyone. Thanks for the organizers for putting this together, because I have been just geeking out to all the talks. Thanks to all the speakers. This is really fun and exciting. I've learned so much already. So. There's some trick.
Oh yeah, click on the screen. Yeah, just click on the screen. There you go. Yeah.
Oh. Great, and then it should go forward here. Okay. So I think we've all probably read papers at some point that have this very common sentence in it, which says, in order to persist under warmer and drier conditions in the future, species have to be able to tolerate, adapt to climate change or move, right, this idea. And it does make sense. So most of what I'm going to be talking about in this particular piece is the potential for this species to tolerate climate change or adapt. But it makes sense to consider those before we think about moving stuff around, right? So this is a figure from now, kind of an old paper, but it shows the likelihood of increasing droughts in the future in the warmer colors, where the stippling indicates where we have pretty high confidence in those predictions. And you can see in the western United States, we really expect to have drier conditions in the future. And I always point out that drought has kind of a specific definition, but regardless, we expect to see rising aridity in the future, because with warmer temperatures, there's greater evaporative potential. So it's really a very significant aspect of global change. So one of the questions that my lab has wrestled with is whether existing genetic variation across species ranges predicts their potential to tolerate and adapt to climate change. And this is because obviously widespread species already experience different climates across their range. So this is a figure of California where the colors indicate an aridity index. In Northern California, of course, it's cool and wet, and in Southern California, it is genuinely warm and dry. And so we expect there to be genetic variation across the species range that's consistent with historical natural selection in relation to climate. And of course, we start with this very simple starting hypothesis that southern populations, which have experienced historically drier conditions, should be better adapted to drought or more arid conditions than northern populations. So here enters Liz Ryan, who was a PhD student in my lab, and we started this project together, and none of it would have happened without her hard work, so give her lots of credit. So this focuses on California poppy, and of course it is our state flower, and it has these it's known for really spectacular super blooms. This is a photograph that was taken in 2019 over Walker Canyon in Riverside County. Did anybody get down there to see that? It was a little bit of a mess. They had to almost close the 15 highway because people just parked on the side of the highway and wandered into the flower fields. But this was, I actually don't know who took this photograph. It was posted on Facebook, and so if anyone ever knows, they have to tell me. But it is really spectacular. It's also a fascinating species because it is incredibly variable across its range. So it's widely distributed. On the map here, the black dots indicate accessions in the California Consortia of Riveria, and then the blue dots are where Liz collected seeds and took photographs of these populations. You can see that there's spatial variation in flower color in the south. In some of the populations, the flowers are very small and yellow. Up here in the north, if you walk around the trails around here, you'll see the flowers are big and orange. There's variation in life history. In the south, they are annual. They live for 1 year. In the north, they are perennial. They produce a taproot. It's like a carrot. It's a big taproot from which they can be perennial. And there's variation in seed dormancy. In the southern populations, one of my populations has 100% seed dormancy. I go out, I put seeds out in the field in a little bag, I pull them out a year later, we cut them open, they're all alive, and nothing has germinated, and there are no seedlings at all. It's really, really strong dormancy. You don't see that much in the north. So, a totally fascinating species. So we collected seeds from a bunch of sites, we grew them in a greenhouse, common garden, under watering regimes that mimicked Northern California and Southern California. And here I'm just going to show you some data from 13 annual populations because we wanted to be able to measure lifetime fitness. But those annual populations went up to about the middle of the state. So this is a figure that's showing total seed mass on the vertical axis, and this is our measure of lifetime fitness. And the populations are arrayed across the horizontal axis, where on the left, these are the populations that are found in the arid part of the state, and on the left is the wetter part of the state. So this is basically south to north. And then the green dots show where we had high water availability, and orange is showing drought. And what hopefully you can really see is that these southern populations are better drought adapted than northern populations. They have higher fitness under pretty low amounts of water. They are also better able to capitalize on high water in order to produce a lot of seeds. Now this makes evolutionary sense. Southern California has the greatest interannual variability in precipitation of anywhere in the United States. So populations that persist in those places need to be able to capitalize on high rainfall years to produce a lot of seeds. So those data, along with a bunch of other data that I'm not going to show you, but I'm just going to tell you, suggest that these southern populations have a suite of traits that are associated with drought escape. They flower really fast. They complete their life cycle while water is plentiful during the winter rains. So that makes them annuals. They have very thin leaves, right? So even though it's dry on the southern end of the gradient, where you might think they have thick leaves, they don't. They have really thin leaves so that they can grow fast, and this is associated with this life history. They don't invest at all in roots. They have just these tiny, wimpy little roots. They're allocating all above ground more towards reproduction, and they produce a lot of small seeds that have high potential for dormancy. So, and that's what produces these super blooms, especially in Southern California, that people get so excited about. Okay, so but what about those northern perennial populations? So in order to get to the perennials, we needed to not plant them in pots. So we embarked upon a field experiment with 20 populations with factorial combinations of temperature and rainfall. These are polycarbonate hexagons that warm up the local temperature about 2 degrees C, or 1.5 to 2 degrees C. And then these are sort of the controls for those hexagons. That's just shade cloth to give them the same amount of shading and sort of protection from herbivores. And we ran it for 3 years because in the 4th year there were only 2 individuals of the over 1,000 individuals left that we planted. So at least in a San Diego common garden, the perennials are short-lived perennials, around 3 years. I'm realizing I didn't put the data slide in here because it was the same, but we found the same pattern where the southern populations were better adapted to drought. But instead, what I want to show you are the trait responses. So on the left, I've got the number of days it takes for a plant to flower, from germination to flower. And on the right, I have specific leaf area, and this is an annoying trait that ecologists love, but it basically means leaf thinness. So a high value means a really thin leaf. And then again, on the left of each of these, we have the populations from the southern dry part of the state, and on the right, we've got the northern wet part of the state. The green dots, in this case, again, are ambient water. The orange dots are drought, and then the open dots are ambient temperatures, and the closed dots are warm. Okay, so hopefully what you can see here, first of all, is that these southern sites flower really fast, much faster than the northern sites. This is a 40-day difference. That's a huge difference in phenology when these are all grown together in a common garden. There certainly is some plasticity in those responses in relation to warming and drought, but these plastic responses are really small compared to this incredible genetic variation across the species range in phenology. Okay, now in those plastic responses mirror the clinal trends. So, for instance, these populations from the arid sites, when they flower faster, they flower even faster under drought. Okay. So specific leaf area, you can see these southern sites have very thin leaves, much thinner leaves than the northern sites. And again, even though specific leaf area is one of the most plastic traits out there in the literature, that plasticity is very small compared to this genetic variation across populations. All right, so what about this question that we started with? So does existing genetic variation across the species range predict their potential to tolerate and adapt to climate change? I would say yes. these southern populations are better adapted to tolerate drought, and their clinal variation, so clinal variation being that variation across the environmental gradient, is mirroring their plastic responses. But I would argue that because that trait variation is across rather than within populations, it means that protecting adaptive capacity actually requires conserving those populations across the range and considering assisted gene flow during restoration. Now, California poppy is abundant and widespread. So I always want to make the point that we are not necessarily concerned about massive declines in California poppy. But I do think it serves as a really ideal model system to study assisted gene flow if we want to do some of the manipulations that people are maybe hesitant to do with rarer species. So, and here are a couple of reasons why. So this idea that we've been able to show that clinal variation exceeds plastic responses to climate, that could be one way that you could screen for saying this is maybe a species that you think about assisted gene flow for. In this case, California poppy has relatively large and connected populations, so that reduces the chances that you're going to have something like outbreeding depression. So this is this figure from the paper that we were all asked to read, where the local population size is here on the vertical axis and the amount of natural gene flow is on the horizontal axis. And so if you have, relatively large populations, and you have some fair amount of gene flow, those are not species that you're worried about for outbreeding depression. Whereas if you have small populations especially and not a lot of gene flow, that's where you might really worry about outbreeding depression for those species. Another thing about poppy is it's an obligate outcrosser. So yes, that makes it very difficult to work with because I have to chase these flowers around in flower with Q-tips all the time in order to get seeds. But it allows it to be a model for other rarer wildflowers that require animal pollination. And of course, it's not a species of conservation concern. But what I forgot to put on here is there's also some really great genomic resources for California poppy. So one of Jay's students is about to publish the reference genome for it. It's one of the species that's included in the California Conservation Genomics Program. How many people have data from that? A few. If you don't know about the CCGP, it's a really great resource. There's 100 species across California for which we have now pretty good landscape genomics. Most of them are animals. I don't know how many of them are plants. Maybe 20. So there's starting to be some reasonable resources. It might be fun to look at whether or not the gene flow patterns that we see in California poppy across the whole range are correlated with gene flow patterns that we see in other populations that maybe have more restricted ranges. Okay, so the last just couple of slides here, I just wanted to add in some new work, so none of this is published. So this is another Organismal Responses to Climate Change grant from NSF. And of course, Jay is a collaborator on this, along with Horace Barrow, who's at the University of Connecticut, he's the species distribution modeler on this one. And one of the questions, there's a lot of questions that we want to ask, but one of the questions I'm really interested in getting at is what is the optimal transfer distance for assisted gene flow? And Ed was talking about drawing things on bar napkins, and this I just drew on a piece of paper in my office and took a picture of it because I didn't feel like making a figure. But the idea here is that if you have some target population and you want to know whether it's beneficial to bring in individuals from other populations, from varying distances away, you might hypothesize that the benefit, and here the benefit is this way, I don't know why I drew it this way, is it some intermediate population? Because if they're fairly close by, there might be some benefit of there being increasing diversity, but you might not really be bringing in any new pre-adapted climate alleles. If they're really far away, that's where you might think about their starting. If there's, if they're really far away and there's no gene flow between the populations, that's where you might start to see something like chromosomal incompatibilities that can cause outbreeding depression. So there might be some sort of sweet spot, some optima that's in the middle, but finding that is kind of challenging, right? So this is one of the things that I currently have an experiment. The plants are about this big, and that's the plan, is to do some of these crosses amongst all of these populations to figure this out. And one more, there's a lot on the slide, but I'm excited about it. So this is work that my postdoc, Stuart Schwab, is currently, I think this is actually submitted. So another way to think about assisted gene flow is to think about what traits you're trying to introduce into your populations. And so one thing that we've done is we've screened these populations for their drought escape strategies. And California poppy, like many species in California, already experience seasonal drought, right? So they all probably have some drought adaptation strategy if they're in the Mediterranean climate region of California. And one of the things that we found is that all of our populations have very consistent plasticity for a few traits in response to experimental drought. 1 minute? Okay, I'll wrap that. So if you think about it, if all of the populations have it and they're all equally plastic for it, that's not a target for assisted gene flow. That's just great. That's just something we know. But what we did find is that there was clinal variation in these drought adaptation strategies, where the ones in the south, I told you, don't have a lot of allocation below ground. They have this escape strategy. In the north, they have this really high root allocation. That is driven by a fundamental trade-off. So where you have these really fundamental trade-offs, that could be a concern for assisted gene flow, because it's not really clear how, if you have mixing between those populations, how they're going to mitigate this really fundamental developmental program trade-off. Okay, so I just want to thank funders and collaborators and see if you have any questions.